Hello, hello, hello. There we go. Now you should be able to hear me. Thank you very much. I had turned my microphone on. I'll bet you can hear me now, right? Hello, hello, hello. Good to see you. Sorry. Technical problems from your knucklehead teacher. Okay, good enough. Thank you very much. So I told you the secrets to the class. I hope you heard them. If you didn't hear them because my mic was off, I am so sorry. Holy cow. Good morning, Anna. It's good to see everybody. Welcome to class today. Hope you've had a great weekend. We are now in week 5. We are 1/4 of the way through this class. I hope you are as excited as I am. The weather is a bit chilly outside, so it's not a bad day to do some school stuff. All right, let's go ahead and get started. So, this week we're going to be talking about chapter 5, sensation and perception. And today we're going to focus on vision. Uh, This week is sensation and perception. We're going to work on trying to understand how human beings take in and process sensory information that they then use to base their decisions and behavior on. We check to see if you received an email from William Cray. Yeah, I sure will, William Cray. I will definitely check to see if I got that email. So we're talking about sensation and perception. This is the area of psychology that you would usually see the subfield of experimental psychology. So if you folks remember from week 1, we talked about experimental psychologists. They're the ones who study consciousness and sensory perception. So if you find this week's stuff interesting, you may want to go into the subfield of experimental psychology. Today, we're going to talk about the basics of sensation and perception. We're going to talk about where in your brain these sensory cortices are. And then we're going to talk about the visual pathway, how information travels along the brain for the visual pathway. And we're going to talk about color vision and organizing our perception. So that's what we're going to focus on today. Let's go ahead and talk about the basics of sensation and perception. Psychology actually starts with sensation and perception. If you remember that fellow Gustav Fechner, I suggested to you he was the first person to measure the mind. If you remember that from exam 1. First, to demonstrate that the mind was systematic and could be measured. He actually was interested in sensory perception. He was actually interested in this thing called the difference threshold. How much does a sensory experience have to change before you can tell it's changed? And so his just noticeable difference is considered the first scientific measure of the mind. So were we getting light static feedback? Go ahead and talk to me about the sound. How's the sound this morning? That's a great question. I am so sorry about my sound. I'm trying to use this microphone and headset because I thought it would give a better sound, but I'm not sure if the sound is working as well as I would like. A little bit of static feedback. Why are we getting static feedback? Okay, well, we have a couple of students hearing things. That's interesting. Maybe it's because I'm using a cheap headset. I don't know. Can you hear me now? Can you hear me now? Can you hear me now? Hello, hello, hello. Oh, wait a minute. Headset. Why is my headset not even working at all? The sound is good for me. It is not that bad. Well, okay. We're gonna have to I'm gonna have to work on that. So I am not sure what the problem is, but I will definitely get it sorted out. Hello. Any better? Any better? God, I gotta figure this out. Okay. Let's see. Headset. Headset microphone. Got that. Why is my headset microphone not working? Crud. I am so sorry, folks. Okay. All right. I will work on that and see if I can figure something out, folks. I do apologize for the microphone sound. Okay. So the area that Gustav Fechner studied was sensory perception, specifically this thing called the just noticeable difference. And his area would eventually become the modern subfield of experimental psychology. Okay, William Cray is explaining this to me in a language that I sort of almost understand. I don't know what a noise gate is, William, but thank you very much. I'm sure you were right. So, we're going to talk a little bit about the different senses. We typically talk about five senses: vision, hearing, smell, taste, and touch. But you know what? Your brain also has other sensory perceptions that it makes. For example, your brain has the sense of balance. Your brain has the sense of pain, which is different than the sense of touch. And your body also has the sense of warmth to let you know when you are too warm or too cold. Okay, so it's hard to say what the problem is, William, and I don't know, so I'll just go on. But the purpose of our senses is to provide information about the environment that can be integrated, evaluated, and used to organize and direct our behavior. So we take in lots of information, we process that information, and that helps guide you through your day-to-day processes. Yes, it's your vision, your hearing, your smell, your taste, and your touch, but it's also a sense of balance. It's also a sense of heat. It's also a sense of pain. Okay. Now, sensation and perception. Let's talk about these as two processes. Your book talks about it, and we need to understand the difference between these two things. Your full sensory experience is composed of two processes: sensation and perception. Sensation is the process of recording sensory information into patterns of neural impulses. We're going to call that process transduction. So, as you are seeing right now, light photons, are coming into your eye. And these cells in your eye, called rods and cones, are turning that light information into patterns of neural activity. That process is called transduction. Okay, the process of turning mechanical energy into neural pathways is called transduction. And when you transduce information, you create patterns of neural impulses. We'll call that general process sensation. Okay, and we have specialized cells that turn physical and chemical energy into patterns of neural activity. So you've got rods and cones, cells, neurons in your eye. You've got hair cells in your ear, inside your ear, in your cochlea that bend or don't bend, that allow you to judge sound. You have taste receptors or taste buds on your tongue that respond to chemical information. You have olfactory receptor cells in your nose that respond to chemical information as well. And then on the outside and the inside of your body, you have pain, pressure, and temperature receptors that are all responsible for transducing mechanical energy into patterns of neural impulses, sensation. Now, here's the trick. The full process of sensory experiences depends upon a secondary process, which we call perception, which is making meaning of these pattern of neural impulses. I might see the color red and the shape round, and then I decide, hey, that is an apple. Okay. I might smell something really sweet and see a brown thing in front of me and go, wow, that is an apple pie. So, sensation and perception are two different processes. You have these receptor cells in your body that turn mechanical information into pattern of neural impulses. We call that specific process transduction, and that leads to the experience of sensations, if you will. We then turn those sensations into full experiences by giving them meaning, and that is called perception. So, sensation and perception. And when we talk about visual illusions, we're talking about our perceptive processes being fooled by the pattern of sensory stimulus that we experience. I've got a, a, uh, a video in your course resources folder about a fellow, Ben Underwood, who can hear with his ears. He turns echolocation, he turns sounds into visual experiences. He perceives vision. Even though he's hearing sound, he's perceiving vision. So we're going to talk about sensation and perception as two processes, okay? That's right. The activation of sensory neurons at the level of the stimulus, okay, which is sensation, and perception is the integration of the sensory information into meaningful patterns, which happens in the central nervous system. Now, I told you yesterday, or I told you several weeks ago, we talked about motor neurons and sensory neurons. Sensory neurons bring information for your body to your central nervous system, and then motor neurons send impulses out from your central nervous system to your peripheral nervous system. There's a third type of nerve cell, neuron, that we're going to talk about called the sensory receptor cell. And these are the specialized cells that change external stimuli into neural action or action potentials through this process of transduction. So we're going to talk about these these sensory receptor cells. You have several different kinds of sensory receptor cells all through your body. You have these things called mechanoreceptors, and they turn physical energy into sensory experiences. Your hearing, you have these hair cells that are bent by air pressure. That's a mechanical process. When you're touched, that's a mechanical process. And so you have these mechanoreceptors that turn that physical touch, pressure, and push energy into sensory experiences. We also have chemoreceptors. Your nose and your mouth use chemoreceptors. They turn chemicals into patterns of neural activity. In your cell, you have visual receptors, which turn light energy into patterns of neural activity. Throughout your body, on the inside and the outside, you have thermoreceptors, and their job is to read the heat on your skin and in your internal organs. And then we have these things called nociceptors, which are different than sensory receptors. So mechanoreceptors that feel pressure are different from nociceptors. Now, nociceptors, in a sense, can respond to pressure. If you cut yourself, that tear in your skin will be felt by your nociceptors, but your nociceptors also respond to chemicals that are released when your body's damaged, right? So these are the different types of sensory receptors. Remember, receptor cells are at the very end of the process, and they turn the physical energy around you into patterns of neural activity that you can then interpret in terms of smell, vision, hearing, touch, taste, and pain. All conscious experiences. If you remember 2 weeks ago, we talked about the thalamus, and I said it was a sensory way station. all of your conscious sensory experiences are processed through the thalamus. This is almost right. All of your conscious sensory experiences are processed through the thalamus except for smell, which probably developed so early that it developed before the thalamus was fully evolved in the brain. Now, if you look at the brain, if you remember from chapter 2, we talked about the primary sensory cortices. That is where information is processed, where information is primarily processed in your brain about your senses. I'm going to be interested in you knowing these sensory cortices. So, in your frontal lobe of your brain, you have your primary olfactory cortex and you have your primary gustatory cortex. These are both in your frontal lobe. In your temporal lobe, you have your auditory cortex, okay? In your parietal lobe, in the front of your parietal lobe, you have your primary somatosensory cortex. And in the back of your brain, in your occipital lobe, you have your primary visual cortex. Now, why do you say primary, Chris, and not just olfactory cortex? Why do you say primary visual cortex instead of visual cortex? Because your brain processes information in a very rudimentary form and then transfers that information to an association cortex that then decides what it is you are experiencing and adds in the final detail, the perception. So your primary visual cortex, your primary somatosensory cortex, your primary olfactory and gustatory cortex and your primary auditory cortex. There, those places are where the sensory information is processed in a primary fashion. Your sensation of the of whatever it is you're experiencing is seen, and then that information is transferred around your brain where meaning is added to it and you create the perceptions of objects. So if you remember from 2 slides over, I talked about the difference between sensation and perception. Your sensory experiences are sensed in these primary cortices. That information is then transferred to other parts of your brain, higher areas of processing, where you make meaning and figure out what it is you are looking at, which is the perception. And visual cortices, visual illusions, right? Sensory illusions are caused by patterns of sensory stimulus that fool your brain into creating the incorrect perceptions. Your brain is fooled into creating the incorrect perceptions. For example, take a look at this right here, screen share. Do you see this? Okay, so do you see this picture right beside me? Do you see these black dots bouncing around the screen? Do you see these black dots? Let's see, right here. Am I showing you the black dots? Does anybody see the black dots bouncing around? Right? Now, what you have here is a gray grid with gray squares, but your brain is actually fooled into thinking that some of these dots are black. And as your eyes move around, that happens. What about this right here? Does anybody see these little circles moving? Do you notice how these circles are kind of moving? Again, this is the sensory experience fooling your brain into adding in perceptive motion. Perceptive motion. So, in a sense, this thing is not moving, but your brain is adding that movement in after the fact. Okay, so let's see. Let's go back to our PowerPoint presentation. All right, so in a sense, that's the difference between what we call sensation and perception. Visual illusions typically occur at the level of perception, where your brain is tricked into creating images that you thought were there. And this happens in your association cortices. All right, so what do I want you to know about sensation and perception? Number one, we have these sensory receptors. They're all around our body, and their job is to transduce, to turn chemical and mechanical energy into patterns of neural activity. That then travels to your primary sensory cortices, where it is turned into sensations. Your brain then interprets and adds information into these sensations to create the final sensory experience that you think of. Oh, I see my friend Fred. Oh, I smell an apple pie. Oh, I hear my mom calling me to come home for dinner. Okay? So I want you to understand sensation, perception, and transduction as very important parts of the sensory process. Now, let's talk specifically about the eyeball. Okay, vision is probably it's been studied the longest. It's probably the sensory experience that we know the most about. And in fact, unlike most psychological experiences, we can actually map the entire pathway of the visual experience in the visual process. We know much more about vision than we do about any of the other senses. All right, so let's talk briefly about the eyeball. Now, let's talk about, let's see, your eyeball. So if you see this picture right underneath me, you'll notice that you've got a sclera and a cornea, which is the covering of the eyeball. And you've actually got a choroid layer, which is the vascularized tissue that includes the blood supply for the eye. All right. Now, and then if you look down, we've got an iris, which is a band of muscle, a colored band of muscle. I have a blue iris. Some of you have brown irises. Some of you have green irises, which is a band of muscle around the pupil, which is the pupil is a hole in your eye. That black dot you see in the middle of your eye is not a black dot. The reason it is black is it's actually an opening into your eye. And the reason it looks black is because there is no light coming out of your eye. If you've ever been to the optometrist and have them shine that light in your eye and look, what they're doing is they're looking into the back of your eyeball. Okay, so the iris is the colored band of tissue that adjusts the amount of light that gets into the eye through the pupil. Because our eyes only work if they have a certain amount of light. If they get too much light, it overwhelms the eye and you can't see anything. If you don't get enough light, you don't get enough sensory information into the eye. So we've got this iris that expands and gets smaller to regulate the amount of light that comes into your eyeball and goes to the back of your eyeball. Now you'll notice that on this picture that you also have a lens, which is a transparent piece of tissue that focuses light on the retina. If any of you have ever had glasses, if any of you have contacts, you have what are called corrective lenses. They are corrective lenses because the lens that you have does not focus light exactly on the back of your eyeball. And that's what corrective lenses are for. They help adjust that light so that when it gets to the back of your eyeball, it is focused perfectly on the retina. Has anybody here ever had LASIK surgery? Anybody ever had their vision corrected through LASIK surgery? When they do LASIK surgery, basically what they're doing is they're reshaping the eyeball so that the light information that comes through your lens is focused properly on the back of your eyeball. So they sort of, in a sense, change the shape of your eyeball just a little bit, and that helps your lens focus the information perfectly on the back of your eyeball. Now, here's the thing. I am now in my 50s, so when I try to read stuff close up, I have to put on reading glasses. That's because the lens becomes less flexible. You actually have these tissues called ciliary muscles, and their job is to bend and flex that lens when you're looking at stuff that's very close up. Your eyes are normally set to see things far off, and when you move up close to something, you'll notice that your eyes start to feel a little bit of pressure. That's your ciliary muscles bending your lens. When you get to be my age in the 50s, this lens becomes less flexible, just like all of your body parts become less flexible. You can't stretch as much. And so you need bifocal glasses because your lens is no longer flexible and your ciliary muscles can't adjust the shape of your lens so that you can see things close up. Okay? When it comes to, does that come after? Ooh, William Craig asks a really good question. Are we aware of the information when it enters the primary visual cortex, or does that come after? We are aware of that information in a sense. However, what you're seeing, William, is not so much, you're aware of what's in the occipital cortex, but by the time it gets into your conscious awareness, it's been changed around and adjusted based on your expectations. So you are aware of what comes into your visual cortex. Well, I take that back. It has to get to your verbal area. Let's say this is, you are aware of what's processed through your primary visual cortex, but that information has to travel through other areas, William, to get to your conscious awareness. Okay, that's a great question. That's a really good question. Now, on the back of your eyeball, you have this neural tissue. Is there a way to see objective reality then? No, no, there really isn't, William. Like I said, you are aware of it in a sense when it goes into your occipital cortex, but you still have to make meaning of it. Everything you perceive, you add meaning to. So there really is no such thing as objective reality that you can perceive. I take that back. There is such a thing as objective reality, but nobody, no human, ever gets to see objective reality. Instead, you get to see the reality that your brain creates based upon the patterns of activity. That's a really good question, William. You're asking the deep philosophical questions today in class. I like that. Okay, now the retina is in the back of your eyeball, and it's neural tissue that contains these visual receptor cells. Remember, I said receptor cells uh, turn external information into patterns of neural impulses. The receptor cells in your eye that do that are called visual receptor cells, and there are two types. They are rods and cones. And if you look at a picture, you'll see that your rod-shaped cells look like rods and your cone cells look like cones. And these are in the back of your eyeball. Now, if you are looking directly at me right now, I am falling on your fovea. Your fovea is where objects fall when you are looking directly at something. Now you'll notice no blood vessels in this little area. So I mentioned this idea, this thing called the choroid. You have not as much blood supply in the middle of your eyeball because it's packed with these rods and cones. And as you move towards the periphery of your vision, you have less rods and cone cells and you have more blood supply. So when you are looking directly at something, you see it incredibly clear. The stuff on the periphery of your vision, you do not see as clear. In fact, check this out. The stuff that you are seeing on the periphery of your vision, you are not even seeing it in color. You are instead seeing it in black and white. But check this out, William. Your brain adds in the color afterwards. So even though my hands are moving in my periphery, right? Now, really all I'm getting is black and white information on my hands. But because I know I'm a Caucasian guy with moderately tan skin, my brain records two tan hands moving around. But actually, I'm not seeing tan hands. They are black and white. So your fovea is where your clearest vision is because you don't have any blood cells. You have nothing but visual receptor cells. And it's where your color cone cells are located. So you really have clear color vision right in the middle part of your visual scene. and everything around the outside is a little bit blurry, and it is also in black and white. Now, the reason that you don't see that is because of this perceptive process. Your brain adds in the visual information that it thinks is supposed to go with the scene that you are seeing. All right. Now, here's the deal. Right, sort of not in your phobia, but to the inside of your phobia, just a little bit, there's this thing called the optic disc. And that is where these nerve cells, these receptor cells, leave your eyeball. And where they leave your eyeball, there are no visual receptor cells to perceive anything. So you have a blind spot in your field of vision. So for my right eye, my blind spot is right out here a little bit. For my left eye's blind spot is right out here. That means that things that fall in my blind spot are not perceived by me because there are no visual receptor cells. You might say, well, Chris, I don't see any holes in my field of vision. You're right. Now, this occurs because of two reasons. Number one, when I have two eyes, although my right eye is blind right here, my left eye has receptor cells that can see that information and fill in the missing information that my right eye doesn't see. You might then say, okay, Chris, I'm going to close my left eye and stick my right hand up here, and I still don't see a blind spot. My hand doesn't disappear no matter where I move it. And the crazy thing is, because your hand this blind spot's kind of small, and when your hand falls on either side of the blind spot, guess what your brain does? It adds in the missing information. Yes, there's a hole right here where I'm not seeing any visual information for my hand. But you know what? My brain knows what a whole hand looks like, so it adds in the missing information. If any of you uh, have ever done the blind spot test, you'll notice that what they give you is a very small X, and you're supposed to move that X. You're supposed to close the eye, and then move that X forward and back. And as long as it's small, if it falls fully in the blind spot, it disappears. But as long as part of that X is outside the blind spot, your brain adds in that extra information. All right. So from this slide, what I want you to know, I want you to know that the iris adjusts the amount of light that gets into your eye that goes to your retina. And in your retina, you have these receptor cells called rods and cones. And these rods and cones are sensitive to stimulation from light. You can't hear me? Can, is my sound gone? Because I'm looking and I seem to have sound here. Can you folks hear me? Yes, no, who can hear me? If you would, please go ahead and type in, let me know if you can hear me. You can. Okay, Dearica, it's okay. All right, Dearica, it's something on you then. All right, good enough. Thank you for checking and let me know, Dearica. Sometimes I'll go on lecturing for 10, 20 minutes without my sound, but in this case, it's you. Okay, now let's talk specifically about this retina in a little more detail. The retina contains two types of visual receptor cells. Visual receptor cells contain proteins called opsins that are sensitive to light photons. Okay, so how do we turn light energy into patterns of neural activities? You have these proteins that are in your rods and cones called opsins. And when light energy comes into your cell, and stimulates this opsin causes your visual receptor cell to generate an action potential. Remember we talked about action potentials from chapter 2? Opsins cause neurons, opsins when stimulated by light cause your neuron, your in a visual receptor cell is a neuron, cause it to generate an action potential. And if you look at the picture right underneath me, you'll notice that we have rods and cones and that they generate action potentials that follow that black pathway. you'll notice that they follow that black pathway and they go out and then they exit through the optic nerve where your blind spot is. Okay, now let me talk a little bit about these types of rods and cones of these types of cells. If you look at the picture over here, you'll notice that your rod cells are long and rectangular and look like rods, and you'll notice that your cone cells come to points, thus the name rods and cones. Now, your cones, let's start with your cones. If you are looking directly at me, I am falling mostly, my image is falling mostly on your cone cells. Now, your cone cells are going to be located almost exclusively in the fovea, and they need lots of light in order to be stimulated. It takes lots of photons to cause a cone to generate an action potential. And these cone cells actually have three different kinds of opsin that respond to three different types of wavelengths. And this is what gives you color vision. So your cone cells are what help you generate color vision. But color vision only works in bright light because these cells need a lot of light in order to generate action potentials. Now, you also have these other kind of receptor cells called rods. Your rods are much more sensitive to light. They take less light to stimulate, and they only have one kind of opsin. It's called rhodopsin. That will not be on your test, rhodopsin. But they contain only one kind of opsin, and that opsin is sensitive to stimulation from any type of light. And so your rods generate generate black and white vision. However, since your rods are more sensitive to light, they allow you to see in low light conditions. So if you've ever walked around your house at night with the lights turned off and you can see your bed, you can see the couch, you can see the shape of the TV, that's because your rod cells are working. Now, the reason that everything looks black and white at night when you're in the dark is because your cone cells are not working. They are not being stimulated at all. So everything you see is going to show up in black and white. But your rods are much better for working in low light conditions, and they are mostly located in the periphery of your field of vision. And these cone cells are visual receptor cells that are sensitive to that are not as sensitive to light, but they generate your color vision. Now, if you look at this picture underneath me, you'll notice that we've got the cone cells, the red and blue cone cells. And then you'll notice outside the red and blue cone cells, we have these things called ganglion cells. Ganglion cells tie our rods and cones together so that they can work together. So whenever a green cone cell is firing and you're seeing green, or let's say green, uh, your ganglion cell is actually stepping on a red cone cell so that you can't see red. If anybody's ever seen one of these things called a visual and after image, if you look at a red screen for a long time, 30 seconds usually, and then turn and look away at a white wall, what you're going to notice is that you see the after image that is the color of green. And what happens is, while you're seeing red, your ganglion cell pushes down your green cell and says, don't fire, don't fire. But when the red goes away, the ganglion cell lets up off of the green cell, and that green cell is going to pop back and start sending a little information. And so you're going to perceive a color after image, right? So if I have you look at blue for a while, and then look at a white wall, you'll see a vaguely yellow image. If I have you look at the color of red for a while and then look at a white wall, you're going to see a green after image. And it's because these ganglion cells, when one cell is perceiving something, the ganglion cell shuts down the cells around it so that you only see that one thing. Now, these ganglion cells, depending upon where you are, they cover more rods and cones. So outside of your fovea, outside of your peripheral vision, a ganglion cell might connect as many as 50 rod cells. But in your fovea, when you are looking directly at something, your ganglion cells connect with your cone cells almost 1 to 1. And this is what gives us very high visual acuity or very clear vision. Okay? And so your ganglion cells are part of the visual process. Check this out. You have 10 million rods and cones in this eyeball, okay? But you only have 1 million axons leaving the eyeball. So the information that comes into your rods and cones is processed and compressed by a factor of 10 by the ganglion cells, so that you only have 1 million messages leaving your eyeball. In fact, the retina is considered part of the central nervous system because this compacting and this processing of information actually occurs in your eyeball. So your retina is actually considered a part of your brain and spinal cord because your retina processes and compacts the information. Okay, people like myself with heterochromia. Nicole, if you would, I'm not familiar with heterochromia. If you could type that in the chat bar, I will explain to you why that happens. Okay, uh, I've good one. Seems like once a week somebody asks me a question that I don't know the answer to. So type in heterochromia and I'll tell you what it is. Okay, now, all right. So color vision. How do we get color vision? Your brain, okay, and let's see, William Cray, I think, was talking about the difference between sensation and perception. All right. Now, your brain, let's see, your eyeball has three different types of cone cells. It has some cone cells, and these three different types of cone cells have three different opsins in them. Remember, opsins are sensitive to light stimulation. When an opsin is stimulated by light, it generates an action potential. Now, in your cone cells, you have three different kinds of opsins. And each of these opsins is sensitive to part of the visual light spectrum. So the light that your brain can process is a wavelength somewhere between 400 nanometers and 700 nanometers. Now, there are some things like X-rays, there are ultraviolet rays. These are all photons, but your eyes are not sensitive to them. Your eyes are only sensitive to light photons somewhere between 400 nanometers and 700 nanometers. Now, with your cone cells, your rod cells are sensitive and respond to any type of wavelength between 4 and 700 nanometers. And that's why your rod cells don't generate colors. However, your cone cells, you have three different types with three different types of opsins that are sensitive to different types of light in the visual spectrum. So you have S cones, which are mostly sensitive to the short wavelengths in the visual light spectrum. And when these cones are stimulated, you see the color blue. Okay, you have these M cones, and they have opsins that are sensitive to light in the middle part of the visible light spectrum. And they give you, when these cones are stimulated, you get the sensation of the color of green. And then you have these cone cells with opsins that are only sensitive to the longest wavelengths in the visual light spectrum. And this sensitivity results in the perception of the color red. Okay. I have two different variations. Pupils with two different colors. You know, no, that's a great question, and no, Nicole, the color of that band of tissue does not have anything to do with your rods and cones. That's a great question because you're saying The eyes are multiple colors. Would that affect my vision? No, it does not. All it does is it makes you a really cool looking person, Nicole. That's all I can say about that. Uh, my youngest son has one eye that is half green and half blue. And I thought that looks pretty cool, but it does not affect his vision. Good question. So rhodopsin is a light-sensitive protein in the eye, cause action potentials to be generated, and that's in your rod cells. But your cone cells contain three different types of opsins: blue cones, green cones, and red cones. Now, if anybody out there is colorblind, you may have what's called red-green color blindness. That's the most common type of color blindness. And what happens in color blindness like that? That's a genetic condition. You are born with it, and your eyeballs don't have either the red or the green option. And so you can't distinguish those two colors. Since you don't have the different cone cells, the different options in your eyeball, you can't differentiate between the color red and the color of green. Okay, and so this is what we would call a genetic color blindness. You're born with it, and it doesn't get any better. And basically, it means that your genes have failed to create one of these options that's going to allow you to differentiate between the color red, and the color green. This is why color blindness occurs in some people. All right. Now, so we've got these rods and cones. You've got this pupil, which is this hole in your eye. You've got the band of tissue, which may be one color, or may, in Nicole's case, may be multiple colors. It opens and closes to regulate the amount of light that comes in your eyeball and hits your retina. When it hits your retina, it stimulates these rod and or cone cells. If it stimulates cone cells, it creates the perception of color. If it stimulates the rod cells, it does not create the perception of color. Your rod cells are more sensitive than are your cone cells. This information that is collected by these rods and cones is then compressed by ganglion cells, 10 to 1, and exits your eyeball through the optic disc, which is known as the blind spot. All right. Now, these axons that exit your eyeball form this thing called the optic nerve. And the optic nerve, remember I suggested to you several weeks ago that a nerve is a collection of axons. And the optic nerve is a collection of neurons that carry information from the eye all the way back to the primary visual cortex in the back of your brain. Now, the information as it travels to the back of your brain, it goes through several places. The first is called the optic chiasm, the optic chiasm. And if you look at that picture right underneath me, you'll see that there's a little crossing area right behind the eyeballs at the front of the brain where your information transfers. Now, here's the deal. Your left eyeball and your right eyeball each see your world. Now, your visual world is divided into a left half and a right half, just like your body is divided into a left half and a right half. And just like I can feel my right arm, and just like I can feel my left arm, I can see the right side of my world and I can see the left side of the world. Here's the crazy thing. Remember I told you how the left side of your brain perceives the right side of your body and the right side of your brain perceives the left side of your body? The right side of your brain perceives the left visual field. It's called the hemifield. And the right side of your brain perceives your left hemifield. So not only do you feel your right and left side of your body, but you feel the left and right side of your visual field. And the right occipital lobe perceives the left half of your visual field, and the right half of your occipital cortex perceives the left half of your occipital cortex perceives the right half of your visual field. Now, the information from each eye needs to be rerouted to the correct part of the brain. So the part of your eyeball that sees your left eyeball that sees the right visual world needs to go to the left brain. And the part of your left eyeball that sees the left part of your visual world needs to go to the right, uh, the right occipital cortex. How does that information switch from eyeball to the correct brain? It does that right there at the optic chiasm. It's the optic chiasm. Now, if you remember from last week, I told you about that structure in your brain called the suprachiasmatic nucleus. Supra means below, chiasmatic means underneath the optic chiasm, and nucleus means it's a bunch of dendrites and terminal buttons. All right, a nucleus is not axons, it's where the communication between neurons happens. So the suprachiasmatic nucleus is located right under the optic chiasm. That's how these brain structures get their bizarre and crazy and long names. Now, most of your eyeball information, I would say 90% of it goes from this optic chiasm and keeps going to the back of your brain. But if you remember from last week, I suggested to you that some of these light cells coming from your eye go to this SCN, the suprachiasmatic nucleus. And that occurs right here at the very beginning of the journey. About 10% of those nerve fibers, those neurons leaving the eyeball in the optic nerve are going to go to your suprachiasmatic nucleus so that you can be tied to the sun, right? And that's what keeps you sleeping at night and staying awake during the day. Then your information goes through to this structure called the thalamus. Remember, I suggested to you that all of your senses are processed through the thalamus except for smell. Visual information stops here on the way to the primary visual cortex. And we're not going to talk about the organization of the thalamus. I'll save that for a biopsych. But it turns out that you have a perfect map of your visual world in your thalamus, and you have a perfect visual map of your visual world in your occipital cortex. And the information comes through and it activates a particular part in this visual map that you have in your thalamus, which then sends an information to the back to your occipital cortex, which also has a visual map. So when one particular place in your field of vision is stimulated, the same place in the thalamus is always stimulated, which then stimulates the same place in the back of your brain in your visual cortex. Okay, so the thalamus is where visual information stops on its way to the primary visual cortex. Now, why do all the senses go through the thalamus? So that your senses can be tied together. For example, I can see my coffee cup and I know exactly where to send my hands so that I can pick up that coffee cup. If I did not have a connection between my sense of touch and my sense of vision, I wouldn't be able to pick up the things in my environment that I can see. And that's what happens in the thalamus. Your senses sort of connect to one another. Now, there's a structure in the back of your thalamus called the superior colliculus. And this is the spot that specifically connects your vision to your other senses. And if you look all the way on the far side of the screen, the two little green balls there on the back of your spinal cord are your superior colliculus. And these are the thalamic structures that allow the integration of the senses. And they connect your vision to your other senses so that when I hear a sound, I know where to look in order to see that sound. The information then goes to your primary visual cortex, where the primary aspects of visual stimuli are processed. Where is that object moving? Where is it located? How is it oriented? And what color is it? So these is all the primary sensory information. And then my brain is going to put that sensory information together in the association cortices. Supra, supra is below, I believe. Suprachiasmatic. I think super is above and supra is below. Supra. Let me see. Definition medical. Above the part. Excuse me. Supra is above it. Supra. Good one. Good one. All right. So it's above it's right above the optic chiasm. Thank you very much for helping me out with that, Rasha. Supra is above. I got that confused today. Holy cow. The students are teaching the teacher today. Pupil, let's see. All right, so we're now at 9:54. I have run out of time because I was a little bit slow and spent too much time running my mouth. So what we're going to do is tonight in webinar and Thursday night in webinar, we're going to talk a little bit about the perceptive process of vision. And so we'll talk a little bit about these gestalt cues and monocular depth cues uh, tonight when we get to class. Yes, thank you, Logan. I appreciate the affirmation there. Okay, do we have any questions? Do we have any questions? I am so sorry to be slow today. I did not get through the entire lecture. Um, Basically, what I'm going to talk about with the organizing principles of vision is what we call the perceptive process. It's the way we put our visual stimulation together in order to make meaning of what it is we're seeing. So your neurons, your rods and cones don't need any training. They work at birth and they provide information at birth that they will provide the day you die. However, what changes as you grow up is you begin to notice patterns in how these visual stimuli come together. And these patterns you learn, and they allow you to begin making guesses about what it is you see in your visual world. And this is the part where your brain can get tricked. So, for example, looking at this picture underneath me, notice these illusory contours. Do you folks see the yellow square in the middle of the blue circles? Well, you know what? That square isn't actually there. Instead, your brain puts that there because that's the shape of a square that you've seen before. And so your brain interprets a square in the middle of these four circles. If you'll notice the picture beside it, closure, you notice that you see the triangle. Well, there actually aren't three triangles. There are three purple V's, if you will, but they create a shape that your brain has learned to interpret as a triangle. Okay? And so, in a sense, there are a lot of habits of vision that you learn early in life that then direct the way you interpret the visual stimuli that you are exposed to. Does that make sense? Okay, we are out of time. Remember, don't forget to come to webinar tonight at 8 p.m. We're going to be talking about visual illusions, sensation, and making meaning in the visual with visual information. So we're going to talk a little bit about perception tonight in class. Does anybody have any questions? There's a circle in the triangle. There's a circle in the triangle. Whoa, a circle and a triangle. That's pretty deep, Logan. You know, now that I'm thinking about it, I see an upside down triangle maybe, or a circle. Wait a minute. Yeah, I see the circle. That's kind of bizarre. Well, thank you, Rasha. I hope you liked the lecture. And thank you, actually, Rasha, for the help with supra versus super. I should have known that. Um, so if nobody has any questions, I'm going to go ahead and log off right now. If you do have a question or want to talk to me, don't hesitate to send me a text, and I'd be more than happy to talk to you about that. Otherwise, I look forward to seeing you tonight at 8 o'clock in class. Thank you, Mackenzie. Folks, thank you. Have a great day. I look forward to seeing you tonight in webinar. Take care.